So yeah, as Philip mentioned, this first session will be on antimicrobial resistance. To talk about it, we have Achim Plum. So he's the Chief Business Officer of Curetis. It's a German company that is working on developing fast diagnostic tests for infectious diseases. You're also the Managing Director of Ares Genetics, which is a subsidiary of this company of Curetis that is searching for biomarkers to diagnose infectious diseases. And I think, you told me you've also worked at Siemens before, also working in diagnostics, before working
in life science research, correct. And just to be very precise here on the Ares Genetics, we have the real manager of Ares Genetics. I'm just a co-manager there. He's sitting here, it's Andreas Bosch, with the team, because we had a workworkshop here in Berlin, so we made use of that.
Oh, cool,
so
yeah, maybe if you have something to add, or if he says something wrong,
really bring it about in Vienna.
You can stop him. Great, so so we're going to talk about antimicrobial resistance because obviously diagnostics is a big part in that. And I guess the thing is, I get the feeling we've been hearing more and more about antimicrobial resistance recently. And there's this figure that I always hear and I always read everywhere. You've probably heard it so many times. Like they say antimicrobial resistance will kill more people than cancer by 2050. I just want to ask you, how true is that statement?
Well, I think it is a very pessimistic view on what's going on. It's certainly based on some facts. It's based on a report, actually commissioned, I think, by the British government originally. I think the industry in general believes that this is probably a little overestimated, but the truth behind is we have a severe problem, and we are losing antibiotics as an effective weapon to fight resistance, and we see more and more multiple drug resistant bugs out there, and up to the point that there are individual cases by now where all treatment options are basically gone. So we had a case in the US a year back or so, a woman that was diagnosed with some severe infections, and out of the 16 possible therapies, 15 didn't work, and the one that could have worked wasn't approved in the US. So that woman died, and in a setting where you wouldn't expect something like that to happen. So high-end US healthcare. It wasn't a kind of sub-Saharan Africa. It was in the midst of the US, in a very well-equipped hospital with all the tools at hand, and that's just the reality. So I think whether these numbers are true or not, it's hard to predict. We certainly try to work against it, so we'll probably never know. It is a severe problem, and it was predicted by the actual discoverer of penicillin at the time, Fleming. I mean, he already predicted that this massive overuse of antibiotics will lead to resistance and make that drug ineffective again, and that's exactly what's happening. And we can't, especially as antibiotics are from a pharma perspective, a tricky business case. If you're successful, you have that great new drug, the best thing you can do it with it, shelve it, don't use it until you really need it, and that's a terrible business model for pharma companies. So you wouldn't expect too much innovation on that front happening. We have to find other ways of making better use of the drugs we have to avoid a scenario that is painted there, that is probably a bit over-pessimistic, but there is some fundamental truth to it.
Okay, so you already mentioned this issue with pharma, but obviously, what would you say is like the main challenge right now to stopping this problem?
I would say that we force by the way if we just look at now the medical use of antibiotics, let's forget about the agricultural use of antibiotics, which is a problem in itself, a huge problem. But in the medical world, we basically prescribe antibiotics without knowing what's going on. So we do it based on assumption and experience, and I'm the last one to blame the doctors doing it because they don't have the diagnostic tools at hand. So at the moment, the typical way of doing it is you take a sample, you plate it on some agar plates, and you wait what growth, and that takes days. And then you put some antibiotics on and say what stops this growth, and then you know after 48, 72 hours what's going on. Now, if you have a severely sick patient in an intensive care unit, probably acquired infection is there for a stroke, a heart attack, whatever it is, and then already kind of fragile in terms of general condition, and then those patients develop symptoms of a very severe infection, probably through ventilation, so deep in the lungs, you have suddenly some bugs. what can you do? You have to start treating. So you do what you believe may work. And we did one of the clinical trial sites when we did our US FDA approval study, just used that as an opportunity to do what we call chart review on 500 patients at their institution. Had nothing to do with our test, just to look at, looking back, how many of those were treated adequately. It turned out only 15% got the right therapy from beginning, 15%, okay, 15. 2/3 were totally overtreated, meaning we create resistances over and over, but we don't help the patient, plus side effects and so on. And then 20-something% were undertreated, and of those, 18 died. And probably 15 could have been saved with an earlier diagnosis. That's just the reality. So we believe that if we find ways to diagnose these infections to the point of not just saying this is pathogen A, B, C, but also it carries those resistances and you shouldn't use those drugs anymore but that drug, or it doesn't carry any suspicious resistances, you can use standard therapy, penicillin or something, ampicillin, something, straightforward, easy, cheap. I think we could solve large parts of the problem. The other one is certainly that about 70% of the antibiotics prescribed by general physicians are prescribed for viral indications. They don't make any sense, but patients expect almost to get some sort of treatment, so they get it. So if we had, and some companies are developing a simple biomarker test, it's viral or not, it would help a lot on the outpatient side. But these are not the severe infections. This is the general cold where people just get an antibiotic to get something. So we have to solve that problem too.
Okay.
But that's not so much our focus.
Okay, so we need to be faster at diagnosing. How much faster do we need to
Ideally, we need to be so fast that the doctor is prepared to wait to prescribe. And that means we have to get it down to the hours eventually. And there are methods to do that. Molecular diagnostic methods can do that. And so I talk to a lot of even intensive care unit physicians that say, okay, even if I have a patient with symptoms, I would be prepared to wait for 4, 5, 6 hours before I start prescribing, if I then can do it in a targeted way. Okay. So, we are getting there. I mean, this is a debate. I mean, you will find physicians who say, well, if I have such a severely sick patient, I won't wait and I'll give some empirical therapy first, but then at least I want to influence the second dose I give, and typically after 8 hours he gives the second dose. And so if I can do that in a more informed way already, I'm already happy. But you find more and more people say, well, I'm also prepared to wait a little and then prescribe the right drug from the beginning. So we have to get it down to we don't have to get it down to the minutes.
Okay. And you mentioned an interesting word, targeted, because we were discussing this concept of precision medicine, which is now very popular in cancer. But I guess in cancer you can just analyze your tumor and then you can select which therapy is the best one for the specific tumor. Can we translate it to fighting antimicrobial resistance?
I hope so. Otherwise, we have a problem with RS Genetics in Vienna. Because that's exactly what we're heading for. So we want to use and basically apply the model that we now see emerging in cancer over the last years with companies like Foundation Medicine and so on, where we really not take a monoparametric approach to diagnosis, but really look at the entire spectrum of genetic changes and then assign a probability of a certain therapy to work and then help to guide therapy. And I think we are technology-wise in a position where we can increasingly do that also for microbial infections. It's very complex a problem. We have lots of bugs and lots of resistance mechanisms, and we have lots of drugs, so compared to cancer. So it's a real data challenge, but we have the tools. So we just need to apply them in a smart way, and that's exactly what we are trying to do in Vienna. So we have currently the most comprehensive genetic database on antimicrobial resistance. Currently around 12,000 isolates all over the world, collected 30 years of history in that, and we profiled them on the vast majority of antibiotics and built some really smart bioinformatics around that to bring those data sets together. And building on that and certainly further evolving that concept, we believe that we are now getting to a point where NGS becomes also fast enough and integrated enough to have sample to answer workflows based on NGS, where the real challenge is not so much the data generation, but the data interpretation. And for that, you will need some reference database with validated information in there, validated biomarkers in there to to run that against the sequencing results and then come to some decision support for the clinician, because the story gets really complex. I mean, if you throw all those markers at a physician, just say, oh, this patient bug has gene X, Y, Z, that's not helpful. You need to come to some score that is easily usable for the clinician with all the regulatory challenges that come along with that.
Okay, and you were mentioning like all these large amounts of data you need to deal with, and obviously right now there's a lot of there's always the buzzword of machine learning, big data. Is this something that you think really needs to be used?
It helps to build the data resource you need for result interpretation. It helps you to mine data that is out there. Eventually, and we increasingly come to the conclusion, when you really want to have a reference database that has diagnostic grade, really with validated results in there, the last step is, the old-fashioned way, really creating the evidence in the wet lab, if you like, and in clinical patient samples, to have that level of validation that also regulators like the FDA would accept. But to focus your work, what are the bugs you want to sequence and profile and so on, you can lose a lot of artificial intelligence approaches to do that in a smarter, more cost-effective way. And so it will be a mix of things. It will be the kind of traditional way combined with some augmented by a lot of artificial intelligence, machine learning and so on. Once you are in the regulated space, we have to see how we can work with self-learning concepts there, because regulatory-wise, that's a very difficult concept, right? I can't go to the FDA and say, well, I can tell you what my algorithm does today, but I can't tell you what it does in 3 months, and they probably have a very clear answer to me, right? So we need to figure out. We'll get there. I mean, this has to be a dialogue with the regulators too. But so it's rather having discrete releases of the database with new content that you generated with the help of AI, but it's fully validated. And basically, we do that already in HIV. We sequence part of the HIV genome. We have a reference database that is updated regularly, and that tells you can use therapy X or Y, but you shouldn't use therapy Z anymore or so. So we just have to scale that up to the level of the microbiology.
Yeah, but it's also interesting that you mentioned regulations, because obviously you build everything, you make it work, but then you need the regulatory authorities.
Yeah. And then you suddenly have to become extremely conservative again. So we have to figure out what is the best way of doing that. We just went through an FDA study with Curetis for the instrument and the first application in the US. So it's a kind of a sample to answer cartridge-based molecular diagnostic systems. We can test up to 130 targets on one cartridge on one patient. And our first panel was for our standards, rather small panel, with just originally 40 assays on it. And this was, just to give you an idea, the most complex submission in that space the FDA ever had. And they put at peak times 14 reviewers on their side against a team of 3 on our side on this to crunch the data. We have submitted 360,000 data points in sliced and diced in all sorts of ways. But just to give you an idea where we are today, and certainly if you now want to scale that up to hundreds of pathogens in one assay, that becomes a challenge, and you probably have to take a kind of a stepwise approach to that. Start with a smaller panel and then get more and more bigger by supplementary submissions and so on. But the FDA, the one lesson we learned in that process is that the FDA is extremely open to dialogue, to really figure out together how to do this. Because I'd say, well, we haven't done it before, so we also have to figure it out, so let's do it together. And that was a very, very good process. I mean, it took a while, but here we are. So we recently got a FDA approval, so now I can say it worked. But it was a very interesting process and very constructive dialogue all along the way.
Okay. Yeah, because I think you mentioned this was one of the first times the FDA was dealing with something like this.
Exactly. I mean, it was the first time that they cleared such a panel for lower respiratory tract infection. That's what they wanted us to call pneumonia in the US. Fair enough. And it was the first time that they cleared so many antibiotic resistance markers on a panel. It was the first ever molecular diagnostic test for Legionella they ever cleared, one of the pathogens. So there was a first in there. The more first you have, the more scary it gets on the regulatory side. So just in the nature of things.
Oh yeah. So, and I guess also with all these challenges getting the technology and then getting the regulations. So in terms of finance, how challenging is to develop something like this?
It's, well, I mean, obviously it cost a lot of money. I mean, just to give you an idea, until we did the IPO in 2015, we raised about 60 million in venture capital. And we then did an IPO, raised 44 million, just did a smaller pipe recently. So, it takes quite a bit of money. And frankly, I haven't seen many companies that get to if they really do an instrument development and application development, that they get there, below 100 million. It's just in nature. But if you compare that to pharma, that's pretty benign, right? The one thing in diagnostic is that the journey doesn't end there. You have to build the market, especially if you bring innovation to the diagnostics market. Things don't sell themselves. You have to create then evidence in the market on the utility and so on. And that, will require some more funding because until you can get to profitability. But that's just the nature of things.
Okay, but I guess compared to the the whole process of developing a new antibiotic, especially if then you're not...
Right, I mean, diagnostics is still a fairly cheap game from a kind of health economic perspective, if you think about it. So, but that's at the same time the shame that even though it is cheap, the preparedness to spend more money on diagnostic is very limited. So especially in, if we look just here in Germany, I mean, getting a new diagnostic into reimbursement is a 15-year journey. And that is ridiculous. Even if you have the greatest evidence on the world, it takes forever. I mean, we focus just on inpatients in hospitals, so we make use of the budget for the patient in the hospital, so we don't have to lobby for separate reimbursement. But if you're in outpatients, until you get a reimbursement code, it takes forever and sometimes unreasonably long, while those diagnostics can provide an immediate, fairly cheap benefit to better treatment. And so, but that's something I think the health insurance schemes have to still learn. So.
Okay, yeah, because it's again you get the approval, but then you have to convince the insurers, and I guess you also need to convince the doctors.
We don't do that. We keep inpatients. We focus entirely on hospitals, and apart from Belgium, every country has a DRG system, meaning you get a budget for the patient with a certain indication. You get a fixed budget. That budget includes some money for the lab work. And we say, well, if you use our test, you may in the end save a little bit of money by getting the patient out earlier. A day less on intensive care unit means 1,500 euro less for the patient. And that sometimes can be the difference between making a margin on a patient, where you have a fixed budget, or losing money on a patient. And so, you basically have to create, with your diagnostics, you don't have to just have to show the medical benefits for the patient, but also show that at the level of the hospital economic system, you either are neutral at better care, that's already great, or you even provide a benefit. And that's you have to show. If you leverage the DRG system, the budget for the patient. If you're an outpatient, that's a different story, because then it's either self-pay or you have to get into the reimbursement scheme, and that is a very lengthy, tedious journey, and the result may not be what you want, because you suddenly may get 10 euro for your test that you spend 100 million on developing. So that may not be the best value proposition for your investors. So.
Yeah. Okay, and then so going back to the initial topic of fighting antimicrobial resistance, because we're talking about diagnostics, they seem to be, I guess, cheaper or faster to develop, but we still need to develop new antibiotics. I would say, is there a way with all this information that we were mentioning earlier that we can inform or we can improve the way we develop antibiotics?
I mean, I guess we have the tools also now to help better or more informed antibiotics. I mean, the one thing you want to avoid if you develop a new antibiotic is develop one that very easily encounters resistance, right? In the normal process of developing such a drug, you do certain tests. So, for example, you passage at a low dose of your new drug and then see whether suddenly you have some surviving clones that acquired resistance. But frankly, that falls a little short, because the only thing you'll see here is kind of spontaneous mutations that confer this resistance, while the vast majority of resistances are probably derived from other pathogens or other microorganisms, not necessarily pathogens. So by some resistant genes, plasmid-based horizontal gene transfer, and suddenly your benign soil bacterium transfers a resistance to a highly pathogenic bug, and you got a problem, right? And that is very hard to foresee in the right, simply phenotypic way you screen for those effects. So I think we have the tools, and we have actually some IP even on that, how to help drug development in early stages, in lead prioritization target prioritization to come to a more informed decision making early stage, and then certainly down the road we can help to actually help to make drug trials cheaper by providing rapid diagnostic for trial enrollment. I mean, this is a big issue. I know of trials where they wanted antibiotic naive patients, inpatients, intensive care unit, ventilated patients. Now, and they wanted, and so this is almost impossible to enroll. No doctor will wait for 3 days to get a result from microbiology whether to enroll a patient that is on a ventilator and suddenly has a pneumonia, right? Nobody. They had enrollment rates of 0.3 patients per site per year. Now that makes trials really expensive, right? So if you had a rapid diagnostic and that is, has a panel targeted around your drug, testing for potential resistance mechanisms, testing for the right pathogens your drug is designed for, and so on, you can increase the enrollment rates. You can help making trials much cheaper, much more productive. You may find more valuable patients simply because you have a solid diagnosis and so on. So there are lots of ways of helping through diagnostics to make drug development cheaper in that space and faster. So, and that's something we also do, and we have done as Curetis. We have provided rapid diagnostics to clinical trials, and we now started talking to pharmas to do this early stage work too. So.
Okay. Then let's see if someone in the audience has any questions for Achim.
Well, I mean, what you do is basically in those trials, you have two arms actually, because the FDA realized that if you just do prospective enrollment, the sheer prevalence of the individual bugs, if you want to have a good panel, you want to cover the usual suspects, and you want to cover those really nasty ones that are rare, but if they are there, you have a problem with the patient. So the atypicals and so on, And that's a kind of a stretch from a statistics point of view. Now, if you do just a prospective trial, you'll never have enough data points on the rare ones, but at the same time, FDA says, we want them on there, we want to help you to get them on this. So what we did is, we basically did about 1,500 patients prospectively, all comers, whoever was, whoever had a sample sent to the lab, basically. So we just took a bit of that sample. We didn't define any other clinical criteria. Just got in, say, suspected pneumonia, please check, and then we took a sample aside. And then the FDA allowed us to collect retrospective samples. Well, they weren't really retrospective, but with known microbiology result. So we weren't blinded to the result, and we could then fill up those gaps that way. And even if we didn't, they wanted basically 50 data points per pathogen, which is a lot for the rare ones. Okay, it's no problem for the usual suspects, but it's a lot for the rare ones. And then they even allowed us to, if we can't get enough of those, to take negative samples and spike isolates from the usual collections into those samples to fill up. So it was a very kind of constructive way to get, very pragmatic way to get to the numbers we needed. That was the basis, basically. But I can tell you the trial design got really complicated because FDA said we don't believe in culture-based microbiology. It's not a gold standard for us. It's standard of care, but not gold standard. So every sample had to be tested with an independent method on top. So and that created these 360,000 data points. So that we had to submit in a lorry. So
the problem of a lack of gold standard, basically.
Exactly. So you have to build your gold standard by a composite kind of. So and that was a basic democratic vote. Your test positive, culture negative, third method positive, you're lucky, third method negative, you have to discount. So that was design, basically.
Yeah. Any other questions?
I frankly, I'm not too familiar. I know that there are some groups working on it, but I'm not really familiar how feasible that is. I mean, certainly we now have a big hype in immuno-oncology, so people certainly then start also, like we try to translate the diagnostics from oncology to micro, so people try that too, but I'm fairly not aware how likely it is that we'll succeed there. We currently dig out a lot of also old approaches, like using phages again and so on. So there's a lot of stuff going on, getting away from small molecule type of antibiotics, but frankly, I'm probably not the best person to ask it. Yeah, the question is, if you then start thinking, let's say biologicals, you could argue that, the likelihood of some resistance developing becomes even, because you have a larger interface between drug target and compound, you know, if you think of an antibody. So that may be distorted even easier by even more possible mutations.
I'm not talking about using antibodies. I'm talking, you know, creating a way to wake up the immune system and target it, the cell. Go there.
I mean, there are some it's also like adjuvant approaches along those lines to kind of combine that and so on. But frankly, I'm not the expert here, so a bit careful. That's something we actually, it's an amazing question, really good question, because that's something we got concerned with too, because we also believe it's not doable as a single company to build and maintain such a database. So we are currently working on a kind of public-private partnership model where we allow the public to really work with those data, give access to a certain point, and through some bioinformatics front end to allow doing research into antimicrobial resistance. The challenging part is then to retain, let's say, the diagnostic use rights of it that we can leverage then to build business and fund the database. We see an amazing number of approaches in the public space trying to, I mean, you find all sorts of data collections. There's this reference center for gram-negatives, and they start to do a bit of NGS, and then some student build a database. Frankly, the student goes, the database deteriorates, and nobody knows how it was programmed anymore. We have seen all this. I mean, you have all these, and what we want to do is really create a platform that is highly professionally programmed, maintained through industry funding, but is made available to the academic and clinical research for research, and then we together can evolve that further. And we're looking at models, how can we make sure that those people who actually also feed data in, keep control over the data and so on. It's really challenging to build those models, but that's a clear objective we have. And we are digging very deep there currently, how to make that happen. And we have lots of discussions with public health organizations and academic researchers on this and so on. But really try to provide some sort of sustainable standard, high-end, better than probably what individual academic groups could produce themselves, and then make that in some sort available for as a joint research platform. There was, I have to say, a company that used that approach to have like pathogen-specific phages that were kind of carried a reporter gene. So the moment they would infect, they would inject a reporter gene. There was some further species discrimination by the promoters of that reporter gene. And so if the cell suddenly lights up, luciferase, or I don't know what they used, and if that bacterium lights up, it must have been bacterium X or Y, because the phage only infects bacterium X or Y. The company is called GeneWeave and was acquired by Roche for very significant money. The problem is, since then we haven't heard of them again, so I'm a bit worried here. So that Roche didn't discontinue their efforts. But at the time when they were acquired, they said they were like 1 year away from the market. But we haven't heard. That is 3 years ago. So I don't know. But they used exactly that type of approach, correct? Mm-hmm. They modified the phages to be species specific, and then, or even subspecies specific, and then use that. The current product that Curetis sells is PCR-based, cartridge-based, sample in, answer out, 4 to 5 hours in between. Currently developing a smaller analyzer that can do it in 45 minutes, but less targets. So that's our current business. That's on the market: Europe, US, Middle East, going for China at the moment. So at RS Genetics, we try to translate that, scale that up to NGS using this reference database approach. We will be platform agnostic, meaning we develop front ends, for sample preparation, library preparation, and back ends in terms of data interpretation, but we don't care what the sequencing technology in between is. So we will not get into the business of, developing sequencing machines and all that stuff. That has been done by others, and we don't want to sink those 300 million into that or whatever it takes. But we'll just be agnostic and just focus on the data intelligence bit of it.
But what's your prediction for using NGS at bedside?
At bedside will take a while. I think we do see that you can already today easily create workflows that provides an answer to the clinician within 24 hours. I think we'll get that then down to overnight. That's, I think, pretty doable with current technologies. Bedside will take a while. And we'll probably also use that as a more initially as a more second line diagnostics. If, you know, the quick and dirty test didn't work, you know, we want to really know what's going on, and then you slowly migrate it from, you know, third line to second line to first line or whatever. But I wouldn't expect it to be at least short-term bedside testing. Eventually, there's no reason, you know, you can do PCR. I mean, we don't say point of care, we say near patient testing, you know, to be a bit more elusive here. But I mean, even if you get those tests, and I think that's the sweet spot actually for those, you know, molecular diagnostic tests, is the little satellite lab on site at the hospital. Not the central lab with all the microbiology, but if you can get it there, I mean, if the nurse can carry it over basically to that little satellite lab that is run 24/7 for the usual, you know, blood chemistry, you know, and hematology stuff. And I mean, our system can be operated by any tired nurse in a night shift. I mean, it's really easy to operate. You just need someone there, you know. So I think these satellite labs will be, and I do foresee that we have get NGS to the integration level that it could also be run in such a satellite lab. And I think that won't take 10 years. It will take 3-5 years to get there. So I'm rather optimistic. Yeah.
Okay. All right, we're running out of time. If you still have questions, you can approach
I'll be
Achim
around, so
later.
happy to.
And thank you very much, Achim.
Thanks a lot.